Okay, great. I think we can get started again. So up next is Eduard Sabido, who is the head of the Proteomics unit at the CRG, Center for Genomic Regulation in Barcelona. And Eduard's topic is again kind of somewhat general one, but a very important one these days, and one that people are increasingly more interested in particular, as Rudy mentioned, the idea of DIA. One of the ideas of DIA and SWATH is to go to very large studies, and for this you need to have good control over how well your system is working. So Eduard, who's also a former postdoc at the group, is going to introduce us to this topic.
Yeah.
Thanks, Eduard.
Okay. Well, thanks, Ben. And as Ben was saying, so now with targeted proteomics workflows, what we are able to do is actually to measure in a consistent way a set of proteins in multiple samples, right? And this has increased the number of samples that we can measure, and also the type of projects that we do. And so this type of new acquisitions allow us to measure large cohorts of samples or patients or whatever, right? And then, so we need to really control each of the steps of the proteomics workflow, because what it happens now is that these acquisitions or these projects do not last for 1 week or several days, but actually it can last for several weeks or months, right? And what we don't want is to go to a data analysis at the end of the project after several months or weeks of acquisition, and then realize that actually our data is not of enough quality and we have to reacquire everything, right? And for this, we need to make sure that, as Tina was saying, right, so that we need to control for sample extraction, but also sample digestion, liquid chromatography, the mass spectrometry acquisition itself, and of course also data analysis. And there have been several community efforts to define common standards and procedures, and there have been different institutions like ABRF in the States, or UPO-PSI, or also and the National Cancer Institute, NIH, PrimexS, or Proteoret in Spain, that have been defining common standards, especially, for example, in types of file formats, but also quality control and quality assurance procedures in order to reduce the variance and increase the reproducibility and robustness of proteomic experiments. And especially active has been the field of quality control in the LC mass spectrometry, right? So in the acquisition part. And there have been several parameters that have been officially defined with control vocabulary, like from UPO-PSI or the National Institute of Standards in the US, so that everyone knows exactly what parameters are measured and what do we mean when we measure this type of parameters. And by measuring these parameters, now the field is or has shifted from a reactive paradigm in which actions were taken when problems occur to a proactive paradigm in which we take preventive actions to avoid the problems to occur. So this is the data quality triangle or pyramid, and in the bottom there are the analytical instrument qualification that answers the question whether the instrument is suitable for the analytical purpose that we want to do, so whether mass spectrometry or mass spectrometers are the correct instrument to do protein quantitation. Then the next level is the analytical method validation, whether the method that we are using is suitable for the analytical purpose that we want, right? So in this case would be SWATH or SRM, PRM. Are they suitable for doing protein quantitation? And there have been several publications actually demonstrating this. So we will not address these first or second layers, but actually we will go into the third and fourth layer, which is the system suitability test that tells us or we want to answer whether the system is working before the analysis, and the QC or quality control checks that is answering whether the analytical system is working during the analysis, right? So while we are performing our experiments. So quality control in proteomics normally is, or let's say it has been several times very subjective. So how many of you have actually checked for your quality control, either yeast or BSA, and then see whether the intensity of certain peptides is of certain value, and actually has asked your partner or your colleague in the lab, say, well, how do you see it, right? Is it okay or not? And just by discussing this introduces already some subjectivity, because this means that there is an objective threshold that everyone can decide and objectively establish whether the instrument is in good performance or whether we have to stop and do some intervention. Also, doing a systematic quality control is quite labor intensive. Most of us have done Excels, extracting all these parameters like number of peptides, number of proteins quantified, PSMs, and this takes, if you do it every day for every second sample, this takes a lot of time actually only collecting this data and then analyzing this data. And what it ends up is actually being inconsistent. So you start for 1 week, for 2 weeks, and then you do it once a week instead of every day, and then you have data matrices of quality control that are not any more useful, right? And having proper quality control system actually is critical for these long-term projects, when, especially in the translational research field, in which we have many samples from large cohorts or from projects that involve different laboratories. So quality control systems need to be systematically performed and evaluated to improve the processes and detect low-quality experiments. We need tools, actually, that are efficient enough to guarantee that the results that we are producing are reliable and reproducible. And also, so eventually, this would need to be computer-assisted and with defined thresholds, right? So that there is an automation needed and that everything runs smoothly without much effort, right? So doing quality control in your laboratories for your experiments should not be an added burden to your current or daily work. So there have been many efforts on this, especially again on the LC-MS system, and there have been many publications offering tools that actually address quality control issues, right? So, for example, Quameter or Simpatico from Karl Mechler, Metriculator some years ago, also an R package, or recently also from Michael McCaws and Brendan. So the AutoQC system integrated with Panorama that actually allows you to do some quality control for certain sets of samples. So let's focus more into the LC-MS system, in which we have a longitudinal type of data. So that data is normally not project-based, but actually one project follows another one, and what we want to make sure is that our systems are working along or working with good performance, not only from one batch to another, but actually during the whole project. And there has been several statistical ways of doing this or approaching this, and one of them has been the process control, process control, in which there is an input, there is a process and an output, right? And this comes from the car industry, right? And the goal here is to early detect any problem that occurs during this process, right? From the input to the output. And here we differentiate spatial causes versus change of variation, and quality is described as inversely proportional to the variance, right? So in this type of approach, there are these control charts which are displayed here. So these are time, the x-axis is time, and here there is certain variable, for example, intensity of a peptide, right? And then there is what we would call the natural variation of the process, whenever it lays between the upper and the lower threshold, and then whenever it goes out of these limits, so we would have a special cause of variation, right? So the common causes of variation are errors associated to the process itself, and for which we don't have a short-term solution, right? So meaning that we should optimize the whole workflow for actually reducing this variance. But then we have the special cause of variation that are errors that can be assigned to a specific cause, right? So for example, let's say some sample is more dirty than the other, has polymer, then the intensity goes down, right? And so here I can pinpoint that something wrong happened, and in the short term, I can do a specific action to fix this problem. Here are the same. So for example, so these are other control or Sheward charts from, for example, peak symmetry or retention time shift, right? So here what we are controlling is that whether the retention type of set of selected peptides is shifting from one run to another during one week period, and whether it stays within the limits of more or less 20 seconds. So depending on whether we have a cause of variation that we can assign to a specific problem, we'll do certain actions, and if we have common causes of variations, so we will do another set of actions, right? So if we can assign a cause, then to improve, we need to find out what changed in our system. So what made, so what was the cause, or what made the, what was the reason why the problem arose, right? Whether it was a concentrated sample, whether it was a sample with too much detergent, whether whatever, right? Whereas in common causes, so to improve, we need to find what to change in our protocol, right? So this is an optimization of our process, right? So there are things that are short-term and long-term. Then if we are able to annotate what are the causes of these problems, what we realize is that most of the QC systems or the quality problems arise or follow the Pareto principle or the 80-20 rule. This means that 80% of the causes are normally associated to 20, or 80% of the cases of nonconformities are associated to 20% of causes, right? And by addressing this 20%, actually we can improve our quality or the quality of our processes. So next is how we do control for this quality. And then we use standard samples, and these standard samples can be of different types depending on whether the type, right? So whether we use the same sample of our experiment. So for example, using so we are measuring a cohort of patients. So for each sample, we can retrieve several parameters, like, for example, number of MS1, number of MS2, number of assigned identifications, and so on, right? So we can use the same samples that we are measuring for the quality control. But also we can use external samples, right? BSA or E. coli or yeast that we measure from time to time during our data sets. Then there is also the source, and these quality control standards can be homemade or commercial. And here there are advantages and disadvantages. Of course, homemade are cheaper, much cheaper, right? So you produce a huge batch of E. coli or yeast samples or BSA, and then you can use it for a whole year, for instance. But in commercial, right, so you also need quality control of your quality samples. And by actually buying them to commercial vendors or from outside vendors, so you outsource also the quality control of these samples. And what they guarantee, or at least if the vendor is of high quality, so you expect them to do quality control on the samples they sell, so that the batch-to-batch variation is minimized. And finally, there is the complexity of these quality control standards that can be low or high complexity, right? And based on this, so some years ago, the group of Carl Mechler introduced the concepts of QC1 and QC2 samples, right? So QC1s were samples of a small amount that very, very well defined, that were measured or intended to be measured in short gradients and several times per day. And QC2, which were defined as highly complex samples that are measured with over or using a longer gradient, and at least are measured once per week. Right, so in this case, QC1s, we could use it for doing the quality control checks, so before and after each sample of interest, and then QC2s, we could use them to do the system suitability test, so before and after each batch of samples. So here there are several layouts on how to do the frequency of evaluation of quality control, and one way would be to do no quality, or at least no external quality controls, right? And the quality control metrics, then you extract them from each of the samples, right? So you can check, for example, whether there are more miscleavage or not in some samples, and then determine whether there was a problem on digestion. Then you can also try to put quality control samples every several samples, right? So like here, every fourth sample, there is a quality control. And then the other extreme is doing a quality control every so every sample, so before and after every sample, right? So that you know exactly how was your system before and after this sample was measured. So if something changed between this quality control here and this quality control here, you actually know that there was a problem with this particular sample, right? So the more you go into this complete scenario, the more sensitive you are in determining the causes of the problems, and the higher the lower the throughput is, right? So here you are injecting an additional sample for every sample you want to measure, right? This might seem a huge investment of time and money, right? So because at least you dedicate half an hour for each quality control, but actually if you think that at the end you are measuring, for example, a project of 200 samples, and the risk is to re-inject everything, right? So maybe you want to spend a little bit more and be more cautious here, maybe every one or every second sample measuring this quality control. So if a problem occurs, you only need to repeat one or two samples, not the whole data set. So now let me introduce you what we are doing in our laboratory. So we are working with two types of samples, the QC1 and QC2, and we have decided to use 25 femtomole of digested BSA that we inject every second sample, right? So we are going into the more complete way. And then we use this for system suitability test, the QC2 sample that we work with 100 nanograms of digested samples, and we have selected these two samples because they are commercially available from several vendors, and we outsource whatever sample preparation is related to this quality control sample. So we buy them ready to inject, right? And in this way, so we rely on the quality control procedures of the vendor. We check them, right? So they send us information about each of the batches, and this is something that actually allows us to really control the quality of all the procedures when measuring these huge batches of samples. So another thing is, well, now that we are generating, let's say, 8 to 10 quality control 1 and quality control 2 per day in our laboratory, so how we can deal with all this data. And so then we started also doing Excel files and extracting all this manually, and at some point we said, well, so we would need to do something that is automated, right? So that allows us to do effortless data collection, that has also an objective evaluation so that we don't have to discuss whether the system is good or right, and at the end that has also a user-friendly interface, right? So to make our life much easier. And then so what we developed is the QCloud, which we have defined as ready-to-use cloud-based quality control system that relies on three main parts, right? So an easy setup, an automated data analysis that includes database search and also for both ID-free or targeted proteomics and shotgun proteomics, and also a friendly user interface based on a web server. So the first part of the QCloud is a thin client that it's developed for Windows. So this is a small software that is installed in the acquisition computer, and it's compatible with different type of vendors and different type of raw data. And what it does is that once you specify the user and the password to the system and what type of instrument you are acquiring your data to, then it looks to the local folder of your QC, so you can store your QC samples in a dedicated folder for QC or in your raw data. So it crawls this and looks for all the raw files that contains, for example, a regex tag called, in this case, QC1 or QC2 in our case. So the software also performs network status checks so that knows whether there is connectivity or not, FTP availability on our server, also checks the status of the raw file so that it doesn't start the transfer of the file if the acquisition is still ongoing. And then it's also responsible for doing the use or to communicate with the server for the user authentication and user authorization. So the server knows exactly who is the user and which is the instrument that is being monitored. So this is the Q crawler. So it selects all these raw files and uploads them into an FTP server. And then we have developed in the server a smooth pipeline that actually integrates or supports both ID-free and ID-based proteomics workflows. So as I said before, including Shotgun, SRM, PRM currently. And then in this pipeline, what we do is we rely on other open source software so that we don't start from scratch or we don't reinvent the wheel, but actually we use the robust MS converters. So here what we did is, so we did several wrappers like the Q converter that knows where the raw files come from. It transforms them based also, it introduces the right parameters, whether the data was centroid, profiled, and what type of vendor was it acquired, and transform all this data into the open format MZML. And then, so once we have created all the MZML files, then we have also done a wrapper of the MS Convert so that all the MZMLs goes into one of the pipelines depending on what is the user, what is the instrument, and what is the workflow that needs to be controlled, right? So, and everything or all this goes automatically. So the user or the operator of the mass spec does not, so once it has been, he or she has defined the specifications at the beginning, does not have to do anything else for processing all this data, right? And at the end of the workflow, what we generate is a QCML plus other files, and then we store some quality parameters in MySQL database, and then we display it through a website, right? So exactly or concretely what we generate are several files like MZML, feature XML, QCML, IDXML. From all these files, we extract all, or at least most of the standard quality control parameters that have been defined by others. And by others, I mean by the community that has discussed which are the right parameters to be monitored. So, and currently we extract up to 23 parameters, and we store all of them in the database. So then it's the third part. So how we display this to the operator, in this case, or the user of the Q Cloud, right? And we do this through a web interface that we have mounted in a LAMP, so Linux Apache MySQL PHP server, and with a front end based on jQuery and Google Charts. And this is what the user sees, right? So actually, this is the page for my lab. So we have 4 instruments. And so here we have the dashboard in which I have, so the 4 instruments that we have in the lab with their status here in green. And I will come back to this later. And then several graphs that plot several of the quality control parameters, like here, for example, total number of proteins or unique peptides along different different days, right? And here there is another plot for peptide areas of a selected set of peptides, also that we can do for several days. So here the data is more dense because this is derived from QC1 samples, for quality control checks every second sample, whereas this is derived from the HeLa sample or QC2 for system suitability test. Not only this, but actually all these plots are interactive with the user so that the user can actually go to one of these points and annotate it with a control vocabulary. So here we know which is the name of the file, when was it acquired, and then we can select of this control vocabulary, for example, to say whether there was a new aliquot of QC, whether the instrument was just calibrated, and in this way, all these annotations are stored together with the quality control data, and then the operator can actually afterwards query all the quality control parameters together with the manual annotations. So in the interface, there are, of course, LC-related parameters like the peak width or the retention time. and also mass spectrometry related parameters, such as, for example, injection time, either at MS1 or MS2, and the mass accuracy or delta mass accuracy between the identified or the identified theoretical mass of the peptide and its observed one, right? And so these are just examples. We have many more plots covering the 23 different parameters. And here what I wanted to show you is some cases that actually occurred to us and how this tool actually helped us diagnostic what was happening, right? So these are a set of peptides that we select from. So these are peptides from the VSA QC1 sample. So we inject it every now and then. So you can see that along the week, actually how it's going down. Then we stop, we calibrate, we change ion transfer tube, and goes up again, right? So, and here is exactly the same for one single peptide or for the injection time, right? And you can see here how it went progressively up until calibration, and then it's restored again. The same happened, for example, in some other weeks with mass accuracy. So instrument was performing, I would say, excellent. Then we stopped for calibration, right? So we calibrate, or an operator of my unit actually calibrated the instrument. Everything seemed fine, but then we restarted the queue, and you can see how this is shaky and how bad this signal is compared to the previous one. But actually, all the checks or calibration steps were actually in green, right? So the instrument told us, well, you did the calibration procedure correctly. And then, so some days later, actually we stopped again, we recalibrated the instrument, and actually we went back to normality. So so out of this, what we discovered is, so this is data from our Lumos. So what we discovered is actually that when we were calibrating positively and then negatively, so the positive calibration was off again, right? So whenever we were calibrating the negative, we had to recalibrate the positive, right? And these are things that actually we realized based on these plots that are now automatically generated in our website. So another parameter is the injection time, also for quality control 1. Here you can see injection times that are going well, and then at some point goes up, and then whenever we see this, now we immediately stop the instrument, clean the S-lens and the front end, we change ion transfer tubes, and then we go back to normality, right? And these are things that actually before, when we were doing everything manually, it was actually, well, hard to first get all this data, hard to then plot it, and then draw some conclusions within hours or days, right? Okay, so for quality control 2, we plot different type of plots. So these are number of total proteins. So here we perform a database search, right? So we know the type of sample that we have. So then we set up a database, and we always use it. And we don't care much about whether grouping or not grouping. I mean, we do some standard workflow. And then what is important for us is that actually these values are consistent in time. So we have number of proteins, number of peptides, and number of PSMs. And then here we also have the total ion current so that we can see whether there is some carryover or some performance issue from one QC2 to the other one, QC2. Then we also check for the median, peak width, right? And whether this remains constant along the week. Also, we check for the resolution of our column. And here, so the resolution is the capacity of the column to separate two peaks. So what we decided is we took two pairs of peaks or peptides that we have very well controlled in our sample. We could take any other combination, and we are actually exploring whether there are other combinations of peptides that are more sensitive to actually or to highlight the performance of the column, right? So, and what we see here is that from time to time, this resolution goes down, and then it's time to change our column. Same for peak capacity, right? So we know that our columns, so we know, and we have very well characterized, which is the peak capacity of our columns. And then what we do is we monitor this over in time. And then when this drops, then again, we know that it's time to change our column. So The other thing is how we set up these thresholds, right? So first, and many of the plots that I showed you had, some had thresholds, some didn't have. And the thing is that for some we know actually, or we have enough data to actually know when is good and when is bad. For others, we are still learning. And for example, and now we started setting two types of thresholds. So one is this one, which is the variable one, and this classifies each data point in three categories. So conformities, if it lays within one standard deviation, within or around the mean. In warnings, in yellow, if it's between 2 and 3, or red, or non-conformities, if it's beyond 3 standard deviations. And these thresholds we set from a period that we know that the instrument was performing well. Other thresholds are hard thresholds, and they come from, for example, the specifications of the instrument or the specifications of the LC system, for instance. So one of the fixed ones, I think it's mass accuracy. We know which is the mass accuracy that we want to achieve with our Orbitrap, and then we set it to I think it's set now to 2 ppms. So, and based on this classification, so we also associate this to specific actions. So green would be continue acquiring, yellow would mean take a preventive action, so that it doesn't evolve to red, and then red means to everyone in our lab, stop the queue and do something to fix it, right? And then there is no discussion on what to do when, but actually the system actually tells us what to do in each occasion. Also this, so for every variable, we have this classification, and then we translate this into the dashboard, which is kind of a summary on the status of all our instruments. And then, for example, in this case, I know that 3 of our instruments are performing very well at the moment, and then the Excel has some problems, right? And the responsible of the Excel would do a preventive action there. So other things that we are working with, right? And this is future work within the Q Cloud. So what we want is actually not only to process and visualize things, but also start interacting with Excalibur so that whenever there is a non-conformity in the system, actually we can put the system on hold and that no samples are lost because of a non-conformity. So, and these are things that we are exploring now with Thermo and the API of their software for controlling the instrument, so that whenever there are problems, we don't continue acquiring samples. Moreover, so we have set this Q Cloud or it has been running for the last 12 months. So now we have it installed, or at least 7 different institutions actually are using it for at least one of their instruments. We have more than 24 instruments in the system, and during this last year, we have processed more than 10,000 QC1 and more than 1,400 QC2 samples in the system. And this actually allows us also to introduce other type of analysis, and which is, for example, now that within within some of the nodes here, we start discussing how to compare the performance of each other instruments, right? So if I have a Q Exactive, maybe you have exactly the same Q Exactive. We are both using the same QC1 and QC2 samples, so how we start comparing it, right? So now we have several nodes that are processing automatically their data through Q Cloud, and then actually what we want to implement and we are working on is to be able to compare yourself with the mean of the others, right? So how are you in the top 10? Are you in the top 50%? Are you average? How is your instrument performing with the rest of the community, right? And then if you want to share your own data with a colleague in Zurich, for example, so that you are able to share this with him or her, right? But otherwise, we would compare always through, let's say, population or community averages. So not only this, but also we did an effort to make this available, so it's very useful for us. But then we said, well, let's put some type of registration so that people actually can also benefit from this. So if any of you would like to do this, so this is the URL. So people need to register. You put your your name and password. Then once inside, you actually define what type of instrument you have. You download the Q crawler, and then you start labeling, injecting QC samples with the right label, right? So QC1 or QC2. And then Q crawler will upload automatically all these files, and you will be able to see automatically the results through the website. Good. So now I've been talking quite a lot on the LC-MS part, but I would also like to address a little bit what we are doing also in sample digestion, so how we control sample digestion. And here again, right, so we can do two types of quality controls in sample preparation. So either do digestions in parallel of known standards, and this is what we call external QC, and then also spiking standards in the sample preparation. And this is what we call the internal sample quality control. And actually, currently we are doing both types. So whenever we do a digestion of, let's say, 20 or 30 samples together, we put one external control. This means a HeLa extract that we digest in parallel. And then we also spike some standards, as I will show you later, in each of the samples to check for each sample whether the digestion has worked correctly or not. So for the external quality control evaluation, so we are checking for each week in which we perform this digestion. So this is week 15 of the year, but actually we would have several plots like this. So how many proteins and peptides we can identify for this particular sample, what is the distribution of missing cleavage for this particular sample, and then we also extract the area under the curve of some selected peptides for this particular sample. And then we can compare all these values from one digestion of week number 1 to week number 2, and so on and so forth. Right? But we also know that there are matrix effects, right? So that whatever it's going well in our external data set or quality control sample, it might not be the case for each particular sample that we are analyzing. So we used these digestive proteins. So this is work from Levert and Virgin Brun within the context of PrimeXS. What they did is they generated a recombinant protein that serves as a scaffold, and then they did a tail of IRT peptides, right? So here they put the IRT peptides. You will see also during this course how these IRT are used for retention time recalibration, and we use this as an external protein that we spike in and control for digestion. So whenever we have a sample from again, right? So from a project, we spike a minimal amount of this protein here, and then we generate the IRT peptide. So, and this allows us to do three things in one, right? So one is to control for digestion. Second is actually generating the IRT peptides within each sample. So each sample has the IRT peptides. And the third is to normalize or to have some references that at least theoretically should have the same intensity if everything went correctly. So in this way, so in QCloud now we also perform or automatically perform the quality control of all digestions, offline fractionation, and also phospho-enrichment, right? And all this data, so instead of calling them QC1 or QC2, we name them like QCP for phospho, 1, 2, 3, depending on the week of the year. And then this allows QCloud to actually upload and process all these type of samples automatically. And then we plot all these parameters that I have been showing to you. So in the case of phospho-enrichment, very fast, so we use a sigma phosphomix, which is a mix of phosphopeptides that we spike again to both the external, so this would be the external digested human sample, so this would be the external quality control for phospho-enrichment, but we also spike it to each particular sample that we want to do phospho-enrichment. And for example, this was an unfortunate case at the end of last year, right? So week 36, 41, 45, and 50, and you can see how our kits were aging, right? So this was a batch of kits that we bought, and then we realized that something was going terribly wrong here, right? And we had to repeat all the all the phospho-enrichments of these wicks. Of course, a part of doing this, so we also generated the standard protocols for each of the procedures that we do in the unit, right? So from calibrating an instrument, or on how to prepare, and how often to, for example, change a quality control sample, right? So every Monday, for example, we change solvents, we change ion transfer tubes, we change the quality control standards. And I mean, this is not a big effort to do these protocols, but importantly, what it gives us is a lot of confidence, right? So questions that before, or we were doubting before, whether, so how long has this quality control standard been in the autosampler, right? So now we know exactly for how long, and everything is traceable, and everything is systematically changed, and this has allowed us also to address much better the problems, because before, whenever we had a problem, the first thing that we were thinking is, oh, I did something wrong, right? Now we know that many things we don't do wrong, actually they are very well done, and that there was a problem for in a particular sample. And in this way, if we go back to this control chart and identifying the problems and how to solve them, so we are now much more efficient on identifying what was the problem causing our non-conformities. And then finally, I want to talk very briefly on data analysis, very basic things that we do, but actually this will be highly addressed in the course. So also all the tutorials are based on data analysis, quality control, a data set that was generated by Stefan Tenzer. And here, I mean, it's very trivial. So the first is to check, or things that we do is whether to check our data distribution. So raw MS data is log normal distribution. This means that if we do log transformation, it should be normal distributed, and therefore we check for each of the samples whether the logarithm of our intensities are more or less following a normal distribution, right? So then we also check for missing values, and we try, I mean, you know that mass spectrometry data is quite sparse, at least on the shotgun approach, but also in targeted approaches like SWATH, due to the statistical and FDR control of peak picking. And then we try to see what type of missing values we have. And then before going to quantitation, so actually try to classify whether we have missing values actually not missing at random, that they are missing for a specific reason, or whether they are missing completely at random, because whether they are in this way or in this other, so how we impute data or how we treat data will be actually very different, because the assumptions that lay behind our imputation methods are very different if we are in this set or in this other set. Right? Another very, very easy thing and trivial thing that everyone should do is actually check whether actually our replicates correlate, right? And here, for example, you can see that actually control number 3 is not so good, and maybe we would like to repeat this injection or actually remove it from our data set. And finally, data normalization. You will have a whole session on this by Olga Vitek, so I will not extend much more on here, but actually what happens with normal acquisition is that actually you have some variability in the intensities, right? And your assumption is that as these samples are mainly the same, so for example, human cell extracts, so that the average intensity of all these samples should be the same. And based on this assumption that you make, actually you do a normalization here. So there are also other types of normalization, so quantile normalization, for instance, in which we don't not only normalize based on the assumption that the mean or median should be the same, but actually that also the distribution, so that the variability of the data set should be exactly the same, because they are coming from the same type of samples, right? But especially important, and especially in targeted proteomics, where you select some target, right? So you select a subset of proteins, and there you select these proteins because you expect them to change, right? And sometimes what it happens, let's say this is a phosphorylated data set in which you trigger a certain signaling pathway, and then it goes up, right? So this is the natural change that you expect, or that you expect in this signaling network, right? And you have selected these phosphorylation targets just because you were expecting them to change. Therefore, if you normalize, right, you actually are removing all the true biological change that you were expecting. So, and with this, what I mean is that data normalization is, or at least the assumptions of that the means should be the same, is, let's say, a correct, or a correct assumption when you work with shotgun, with an untargeted approach. But when you start working with targeted approaches like the ones that we are explaining in this course, so then this assumption that holds in shotgun might not hold anymore in your data sets. So you have to be very aware of what are the assumptions that you do and how you selected your proteins. Because if you selected them expecting to change, then do not normalize. Right, and with this, so I would like to end, right? So emphasizing again, so how important is on targeted proteomics workflows to actually do all this quality control at the different levels so that you can deal with this large amount of samples in large scope applications without having to repeat or everything just because at the end you realize that something went wrong during your acquisitions. Okay, so there is some reference bibliography, especially from Michael Berreman, Vodmir Tremier, and Peter Pichler from the Mechler Lab. So these are reference papers that for those who are interested in quality control actually are nice further reading. And finally, I would like to thank all the people from my group here, especially Cristina Chiva and Roger Oliveira, who have been leading this project. Then also the Core for Life community, which is a set of proteomics unit in Europe that actually we meet frequently to exchange protocols, best practices, and actually they have been beta testers and providing a lot of feedback for the QCloud system. And then also the OpenMS community that help us in all the questions that we had for setting up the pipeline. And of course, so Ben Collins also organizing here the course at ETH Zurich. And thank you for your attention.
Great talk. Just had a question. So you have all this information from these 7 institutes now. So in those 7 institutes, what do you see has become like the main, or like is there a particular problem that you see that's coming up more often?
So I can talk about our data, right? So, and this is something, right? So that we are now, of course, for example, in this particular setup. And so we are hosting the server, so the QCloud. We think that in this way we reduce the workload of the operators to set up the pipelines, to set up which metrics to measure, everything. But then we also have this privacy issue on the data, right? And so far we went through, or we decided not to touch anyone's data and not to mine it without their permission. Right? So I don't know anything about others' data. In our case, most is charging effects, and that we see periodically over the week. Right? And then when we clean, and we do the routine cleaning maintenance every Monday or every second Monday, so we see how it improves.
Maybe, go ahead.
Just a small technical question. How fast is it? So if you run to see one sample, for example, it uploads it automatically after it's finished, and then how fast you get the results?
Yes, so this is a very good question. And so we are below 1 hour. So since it finishes the acquisition and it's plotted in the website. And this allows us to actually do something before the next run is finished, right? And in this sense, so we cannot avoid losing the next sample, but actually we have a quite fast response.
Maybe I can ask one. So we had some data recently, which was a bunch of SWATH data from different labs, but from the same sample. And one thing that I found interesting about it, so there was different performances, of course, between the different instruments. And I noticed you were using, one of the metrics is some absolute signal intensity metric, right? But something we found actually was that this is not necessarily the best predictor of performance. So we had also data where the absolute signal intensity is lower, but they identify more peptides, and maybe a signal to noise metric might be more. Or do you have any experience with this? Or?
Yes, so you're right. And also we noticed with the different instruments in our laboratory. So we are also not 100% sure which will be the most informative parameters of the ones that we are collecting. So actually, we are collecting more parameters than the ones that we are plotting or using for the classification of whether the instrument is performing good or bad. We aim that by collecting all these, right, at some point we will be able to do multivariate analysis and then select the most informative features.
Great. One more?
You know what, you said that the setup that you use at the moment in your lab is to do a QC1 after every sample, right?
Yes.
You said this is quite
Yes.
time-consuming. But we are doing something similar just also for blanking the samples, right? To have always blanks in between. So what's your experience there? And then it wouldn't be such a waste of time to make a QC if it's also for the purpose of washing the column, and how important is it?
Well, so what we notice is that, so you are completely right, so that it has both purposes. So our QC1, it actually avoids carryover to the next sample. And what we see is that from time to time, we have quite a lot of carryover in the QC1. This is also why sometimes the total ion current of our QC1s goes a little bit up, but we actually don't mark it as a negative because we know that it's coming from the previous run. But it's true that this helps us to actually get more consistent total ion currents from replicate samples, right? Because we inject kind of a blank between each sample, and a BCA is almost as a blank.
Okay, great. Any more? Okay, thanks very much, Eduard.